Hello guys, so welcome back. So previously we were discussing that there are some organisms which are urease positive, there are organisms which are catalase positive, right? So in this session we shall be discussing the organisms based upon the hemolysis. Okay, so this slide which represents here, so all these three alpha, beta, gamma which you can see here, all of these things represent hemolysis. So what do you mean by hemolysis? There are basically, I'm sure you heard of these terms like alpha hemolysis, beta hemolysis, and gamma hemolysis. So what is this alpha hemolysis, beta hemolysis, and gamma hemolysis? Is that, for example, look at the beta hemolysis. What I have done is that I have taken an agar plate. Okay, in this agar plate I have added sheep RBC. After adding sheep RBC, what I have done is that I have taken a spatula like this, and then with this spatula I have taken beta hemolytic bacteria, and I drew this in the form of a beta. It means that letter beta is completely having that beta hemolytic bacteria. Okay, so now look here, what is happening is this beta hemolytic bacteria releases a toxin called as beta hemolysin. Okay, so this beta hemolytic bacteria, this beta hemolytic bacteria releases a toxin, and this toxin is called as beta hemolysin. And what is this beta hemolysin? What does it do? What does it do is it will break down all the RBC. Now what exactly I'm trying to teach you is that, for example, let us say this is the beta which I have drawn, right? So this is the beta. Let us say that this is the beta. Now surrounding this beta, what was there surrounding this beta? There are many number. Let us say there are 100 RBC surrounding this beta, because this beta hemolytic bacteria right now are located in the container, I mean the agar plate. So surrounding the beta we have got many RBC. What did I tell you? And I also told you that this beta releases a toxin called as beta hemolysin. So this beta hemolysin, what does it do is it will spread all the way like this. It will spread all the way and destroy all the RBC. It will break down all the RBC. It will cause lysis of all the RBC. So when it will cause lysis of all the RBC, do you think are there any RBC going to be surrounding this beta? No. So obviously there will be no RBC surrounding this beta. So in the picture you can see here, is there any reddishness surrounding the beta? No, there is no reddishness surrounding the beta. So can I tell that not even little bit red, right? So I can tell that this beta hemolytic bacteria are a group of bacteria which will break down all the RBC, which will cause hemolysis of all the RBC. So beta hemolytic bacteria are also called as complete hemolysis. Beta hemolysis performs complete hemolysis. So this type of hemolysis is called beta hemolysis or complete hemolysis, where all the RBC are broken down. Because surrounding the beta you can see a bright vibrant zone, right? So this zone has no reddishness. It means this zone has got no RBC. Now compare beta with alpha. When you compare beta with alpha, look at alpha here, right? So look at alpha. In this alpha, surrounding the alpha, do you think it is more pale or less pale? Obviously it is less pale. When you compare with beta, surrounding the alpha, the paleness is very less. So what do you mean by paleness is very less? What do I mean by paleness is very less is that alpha hemolysis. Alpha hemolysis. Surrounding the alpha, the paleness is less. So what do I mean by paleness is less is that previously what did I do? Surrounding the beta, I placed 100 RBC. Out of 100, all the 100 RBC have been broken down. There is lysis of all the 100 RBC. But here in alpha hemolysis, surrounding alpha, if I place 100 RBC, out of 100, alpha hemolytic bacteria can break down only 50 RBC. So remaining 50 RBC will be there itself. It means the zone of hemolysis or the paleness will be more or less. Obviously it will be less. Less hemolysis, less zone of paleness, and less vibrance or less paleness, right? So this type of hemolysis, can I call it as partial hemolysis? This type of hemolysis is also called as partial hemolysis. On the other hand, look at alpha. Which color is it? Look at alpha hemolysis. Which color is it? It is green in color. So can I tell it is also called as green hemolysis? Yes, this is also called as green hemolysis. So alpha hemolysis or partial hemolysis or green hemolysis. On the other hand, you have encountered gamma. Now compare alpha, beta with gamma. Surrounding this gamma, letter gamma, right, surrounding this gamma hemolytic bacteria, do you find any kind of paleness? There is no paleness, right? So there is complete reddishness surrounding this gamma. It means surrounding gamma hemolytic bacteria, if you put 100 RBC, out of 100, not even a single RBC is hemolyzed, or not even a single RBC is broken down. So can I tell that alpha, sorry, not alpha, it is gamma, right? So can I tell that gamma hemolysis is also called as incomplete hemolysis? Gamma hemolysis is also called as incomplete hemolysis because there is no breakdown of any RBC. Okay, so in this case, tell me, where will I have more hemolysis? For example, if you wanted to grade these bacteria based upon hemolysis, so obviously beta hemolytic bacteria performs more hemolysis than alpha than gamma. So next important thing is, where will I have more anemia? Anemia is loss of blood. Where will I have more anemia? So anemia will be in the beta when compared to alpha than gamma. Okay. Next. So what are the examples of these bacteria? Let us say alpha hemolytic, right, beta hemolytic and gamma hemolytic bacteria. Now when you come to alpha hemolytic bacteria, it is also called as a green hemolysis or incomplete hemolysis. What are the list of bacteria are? One is Streptococcus pneumoniae and Streptococcus viridans. Okay, so when you come to beta hemolysis, which is also called as complete hemolysis, so one is your Pyogenes and second one is A galactiae. When it comes to gamma hemolysis, the first thing is Enterococcus. Enterococcus performs gamma hemolysis. So this is pretty important thing which you need to know for sure. Okay, right. Cool. Now, after this is one of the high yield thing, guys. This is one of the very high yield thing. I want all of you to remember this. Okay. Next, switching on to the next part in the introduction of bacteriology, we should discuss what are the different types of bacteria, right? What are the different types of bacteria which produce pigment, right? So, can I put a heading as pigment producing bacteria? So when you look at the pigment producing bacteria, guys, so what are the different types of pigment producing bacteria which you have? Yes, the first thing, actinomyces israeli. What type of pigment does it produce and what is the color of that pigment? So actinomyces israeli, in Israel the sand is basically yellow in color, so the pigment which it produces is an yellow pigment, right? So that pigment is called as yellow sulfur granules. So let me, I think it's not obvious. So yellow sulfur granules. So the pigment which it releases is yellow sulfur granules. Clear? Now the next bacteria is Staph aureus. Why you call this as Staph aureus? What do you mean by aura? Aura means gold. Aura means gold. So gold is which color? Obviously gold is yellow in color. So the pigment which it releases will also be yellow, right? So even Staph aureus is also produces yellow color pigment. Third important thing, Pseudomonas aeruginosa. Why do you call it as Pseudomonas aeruginosa? What do you mean by a term called as aeruginosa or arugula? Right? So arugula means green. Okay? So arugula you call it as green. So that is why this bacteria is called Pseudomonas aeruginosa, because the pigment which it releases is green color pigment. Okay? So the pigment which it releases is green color pigment. What makes this bacteria to release green pigment? So exactly, if we go little bit in depth, there are two MCQs which you can cover here. What are these MCQs are? This green color pigment is because of two important things. One is called as pyocyanin and another one is called as Verdin. Pyocyanin and pyoverdin, both of them are responsible to impart green color to the bacteria. So whenever you see, let us say necrotizing fasciitis, right? So necrotizing fasciitis is all the fascia will be inflamed, necrotized. So in those cases, guys, after 1 week or 2 weeks after dressing, when you look at the legs, right, when you remove the gauze pads and finally when you look at the leg will almost be green in color, because this Pseudomonas aeruginosa contaminates there and releases its pigment. If you want, you can look. So, right, in infected wounds, you most commonly find this Pseudomonas aeruginosa. So, these are some of the important things which you need to know regarding the pigment producing bacteria. Right. Now, apart from this, another important and high yield stuff which we're going to discuss is that what are the virulence factors which are released by the bacteria. So, these are bacterial virulence factors. So what are the different types of bacterial virulence factors? So one bacterial virulence factor is IgA. One virulence factor is IgA. And where do you find this IgA? Yes, all of you know. You find this IgA in the saliva secretions. So basically you found in any kind of secretions, even in the tears also you will find it. So found in secretions. They're also found in mucus surfaces. They're also found in mucus surfaces. So if any bacteria is trying to enter through my mouth or through my nose or through my any kind of mucus region, right? So the first line of defense will be what? It is IgA. So IgA is the first line of defense. IgA is the first line of defense in my body, which is responsible to not let the bacteria go into my body, right? So all of you know this, right? So IgA is normally present in you and me. I'm not telling this back as I kept the heading as bacterial virulence factors. It doesn't mean that IgA is one of the virulence factor. No, IgA is not a bacterial virulence factor. I'm just forming the basics. The basics are that there is something called IgA, and this IgA is found in your IgA antibody, which is found in your secretions, which is found in your mucus, right? So that is why this is also called as the first line of defense. The first line of defense. Now, so what is the bacterial virulence factor? The bacterial virulence factor is not IgA, guys. In fact, it is IgA protease. What do you mean? What do I mean is that bacteria releases, for example, in my mouth, right now there is IgA normally, and now I have taken the food. When I've taken the food, bacteria entered into my mouth and bacteria released this IgA protease. Now this IgA protease, what does it do is this will cause lysis or breakdown of IgA. So when it will cause lysis and breakdown of IgA, will I have any more the first line of defense in my body? No. So this is the virulence factor of the bacteria. This is the virulence factor of the bacteria. So bacteria which releases, what are the list of bacteria which releases this IgA protease? So this one is virulent. This one is virulent. What are the bacteria which releases this? Examples are One is Streptococcus pneumoniae, and second thing is Haemophilus influenza B, and next thing is Neisseria. Okay, so how do you remember this? How do you remember this is by a mnemonic SHIN. You know what is SHIN? S stands for Streptococcus pneumonia, HI stands for Haemophilus influenza, and N stands for Neisseria. Fine, cool. So you told me that IgA protease is one of the very important virulence factor which is released by the bacteria. Yes, exactly. Now, what is the second virulent factor which you can tell me? The second virulent factor is protein A. So protein A is the second virulent factor which you can see in the bacteria. So what is this protein A? What is the use of this protein A? If for example, this is a bacteria, guys. This is a bacteria. Sitting on the top of the bacteria, there are these blue color proteins which you can see. These blue color proteins are nothing but called as protein A. So what is the use of this protein A? What is the use is, let us say this is the phagocyte. This is my phagocyte in my body, and this phagocyte is very slowly approaching. Let us say that this phagocyte is slowly approaching this bacteria. You see, phagocyte, I've seen it from the long, and now phagocyte is slowly approaching the bacteria. Now as the phagocyte is slowly approaching the bacteria, this protein A will kick the phagocyte far away. So phagocyte will go far away then, right? It means, can I say that it is preventing phagocytosis? Yes. Protein A prevents phagocytosis. So what is the function of protein A? Protein A prevents phagocytosis. Cool. What is the third important virulent factor which you can tell? That is M protein. What do you mean by M protein? What do I mean by M protein is that M protein is also located on the surface of the bacteria, right? If this is the bacteria on the surface, there are M proteins. Fine. So what is the important thing about this? The important thing about this is, guys, now listen very carefully. M protein, look now. This is a bacteria, for example. On the surface of the bacteria, I have got these red color molecules, right? These red color molecules are nothing but M protein. Okay. Now, in my heart, not only that, even in my joints, let us say this is my joint. So not only on the heart, but even in my joint capsule, even in my joints and heart. So in both of these things also you have got these M proteins. So normally M proteins are present even in your joint region, right? They are also present in the bacteria, on the surface of the bacteria. For example, this is my body, right? And if this bacteria is trying to enter into my body and finally it enters, for example, when it enters, my antibodies, what do my antibodies think is that this is M protein and this M protein is present on the bacteria. M protein is also present on the heart and even on the joints. So your antibodies like IgG and IgM antibodies, they get confused because M protein is located on the bacteria, M protein is located in the heart, as well as M protein is also located in your joints. So instead of attacking the bacteria, instead of attacking the bacteria, they go and attack your heart, they go and attack your joints. I'm not telling that they won't attack the bacteria. Obviously, they will attack the bacteria. In addition to that, they will also attack your heart and joints. So when it will attack your heart and joints, what will happen? You will have endocarditis. If it attacks your heart, you will have endocarditis. If it will attack your joints, you will have arthritis. And the last important thing is you will also have acute rheumatic fever. You will also have acute rheumatic fever. So these are the three important things which are happening because of what? Because of M protein. So this type of phenomena, right? This type of phenomena is also called as molecular mimicry. This phenomena is called molecular mimicry. So if I ask you a question that out of all the virulence factors of the bacteria, which virulent factor plays a phenomena of molecular mimicry, then it will be your what? M protein. So this is one of the important thing which you need to know. The last important virulent factor which is released by the bacteria, that is called as injectosome. That is called as injectosome. What is the other name for injectosome? Other name for injectosome is type 3 secretory system. The other name for this is type 3 secretory system. Injectosome is also called as type 3 secretory system. Now what is this injectosome? What is this injectosome is that, first of all, look at the structure of injectosome, guys. This injectosome has got these things. Look here. This injectosome has got a sharp point like this, right? I label the names later. Next, it has got something like this, right? After that, it has even got like this, and finally this is the thing. This entire thing is located on the bacteria, and this is called as injectosome. Now this needle like projection which you can see here, right? So this is called as needle. This is called as needle of the injectosome, right? And this part which you can see here, this one, the blue color part, this is called as outer membrane. This is called as outer membrane. And next, this one is called as inner membrane. So when if there is outer membrane, there also will be inner membrane. Inner membrane means that is plasma membrane. So previously in the part 1, I told you that between outer membrane and inner membrane, whatever region is there, right? So what is that region supposed to be called, guys? So between the outer membrane and between the inner membrane, the region that is present, this is called as what? This is called as periplasm. Periplasm, right? So but anyways, one is called as outer membrane, this is called as inner membrane. Connecting the outer membrane and inner membrane, this part which you see here, this is called connector. Connector is located in periplasm, and connector is made up of obviously peptidoglycan. I have taught you previously if you remember, right? So this is an injectosome. So what is it going to do? What is it going to do is that, for example, this entire thing is the bacteria here. This is a bacteria, and bacteria has got cytoplasm, right? So in the cytoplasm there are toxins, there are different types of toxic proteins, there are virulent factors. All of them are secreted within this cytoplasm, right? They're secreted within this cytoplasm. For example, this entire thing is an injectosome. Okay, this is the cytoplasm, this is the inner membrane, this is the outer membrane, and my finger, this is called as a periplasm, and this is the tip or the needle. So what is happening is this tip enters into my body. This is the host cell, this is my body. So this tip, with the help of this injectosome, it is entering into my body. Now what will happen? This is the cytoplasm. You look here, this is the cytoplasm. So with the help of the cytoplasm, what is it going to do? I'm putting the arrows now. All the toxic proteins, they slowly go up like this. They enter into the needle. They enter into the needle like this, like this, like this, as well as like this. And finally, they enter into my body. So injectosome is basically responsible to inject what? To inject toxic proteins, bacterial virulence factors, as well as the different types of toxins, right? So this is it, guys. So these are some of the bacterial virulence factors. Now, we'll close this discussion by discussing another important thing, that is the differences between exotoxin and endotoxin, because this has been asked quite many times. So you should know the differences between exotoxin and endotoxin. What is your concept of exotoxin and endotoxin? For example, let us say this is your bacteria over here, and let us say this is another bacteria, right? So this is another bacteria over here, and this is one bacteria. Now this bacteria, where I've written exotoxin, it has got some toxins inside itself, inside it. So what is it going to do? Obviously it will release the toxins, and these toxins will eventually enter into your bloodstream. These toxins will eventually enter into your bloodstream. So these are called as exotoxins. They are coming out of the bacteria. Exotoxins are coming out of the bacteria. On the other hand, when there is a concept of endotoxin, you see toxins embedded within the cell wall of the bacteria. Toxins are embedded within the cell wall of the bacteria, right? So only and only when you break the cell wall of the bacteria, these toxins are released. When you break the cell wall, obviously these toxins will be released. Fine. So where do I find exotoxins? Exotoxins are found in both gram positive as well as gram negative bacteria. But where are endotoxins formed? Endotoxins are formed only and only in gram negative bacteria. Are you sure? Only and only in gram negative, not even a single bacteria of gram positive you find endotoxin. There is an exception. Exception is that there is one bacteria, that is Listeria. In this Listeria you have got in this Listeria which is gram positive, there is endotoxin release. Just Listeria is a gram positive bacteria. It is the only gram positive bacteria where the endotoxin will be released. Other than this Listeria, remaining all gram negative bacteria only, there will be release of endotoxin. Okay, so this is the only gram positive bacteria where endotoxin will be released. So this is an exception. Point number two is, let us discuss about the exotoxins in the next session. But what is the important thing is, we have to discuss very importantly about endotoxins. Why should we discuss about endotoxins? The reason is that, for example, if endotoxin is released, right? Let us say that this is a bacteria over here, right? This is a bacteria over here. This is a bacteria, and what is this bacteria consisting of? Yes, this bacteria is consisting of these red color molecules which I'm drawing here. This is called as endotoxin, right? I told you endotoxin is embedded within the cell wall. For example, if I'm breaking the cell wall of the bacteria, what is happening? If I'm breaking the cell wall of the bacteria, the endotoxin is released outside. And what does it do when the endotoxin is released outside? Look very carefully. This endotoxin activates one of the phagocyte in my body, that is called as macrophage. Let us say this is my macrophage. So this endotoxin will activate my macrophage. So when my macrophage has got activated, my macrophage releases three important things. My macrophage secretes three important things. What are these three important things? One is interleukin 1 and interleukin 6. Interleukin 1 and interleukin 6. Second one is tumor necrosis factor alpha. And third thing is nitric oxide. So what are the three important things? Interleukin 1, interleukin 6, tumor necrosis factor alpha, and nitric oxide. Fine. There is a bacteria. Endotoxins are located within it. Let us say I have given broad spectrum antibiotics. When I give broad spectrum antibiotics, these antibiotics caused breakdown of the bacterial cell wall. So when it caused breakdown of the bacterial cell wall, the bacterial cell wall is embedded with the endotoxin, and that endotoxin is released outside. That released endotoxin will activate your macrophage. When it will activate your macrophage, it will release these three things. This is so far we have discussed. So what is the next thing that is happening? The next thing that is happening is, look here very carefully. This is your blood vessel. What is basically blood vessel lined up, guys? Blood vessel is lined up with these cells, and these cells are called as, yes, these cells are called as your endothelial cells. Your blood vessel is lined up with these cells, and these cells are called as endothelial cells. So what? I'm drawing one endothelial cell down here. So this is the blood vessel, right? This is my blood vessel, and I'm drawing one endothelial cell here. So this is one endothelial cell. Within the endothelial cell, I have got an acid that is called as arachidonic acid. Arachidonic acid. What does it normally do? What does it normally do is, arachidonic acid is basically converted into what? Prostaglandins. Arachidonic acid is basically converted into prostaglandins. And what is the function of these prostaglandins? Prostaglandins perform two functions. One thing which it does is that, look here now. So from each endothelial cell, prostaglandin is released. Each endothelial cell, prostaglandin is released. Each endothelial cell, prostaglandin is released. So this released prostaglandin, what will it do is that it will cause dilation of the vessel. That is the reason, guys, whenever you get hurt, what is happening? You see bleeding. Why there is bleeding? Look in the picture. Why there is bleeding? Because, yeah, your blood vessel, your small blood vessels, they get ruptured. So when they get ruptured, even these endothelial cells also get broken down. So these broken endothelial cells, they release prostaglandin, and prostaglandin will cause dilation of the vessel. That is why you see swelling on your hand at the site of injury. Okay, so what is this prostaglandin doing? What are the functions of the prostaglandin, guys? The first function is, yes, the first function is vasodilation. So when my vessel will dilate, right, when my vessel will dilate, the pressure within the vessel will fall, or it will also increase along with the dilation. Obviously the pressure will fall. So when the pressure will fall, when the tension is less, can I call it as hypotension? So prostaglandins directly can cause hypotension. Second important thing is, obviously when you get hurt, you will have fever, right? Why? Because prostaglandins are also responsible for causing fever. So with the help of interleukin 1 and interleukin 6, what is going to happen? Let me tell you. So this is the normal process which I told you, right? So to convert arachidonic acid into prostaglandin, you need an enzyme that is called as cyclooxygenase, which is called as COX, cyclooxygenase. Now, this interleukin 1 and interleukin 6, what does it do is, it all the way comes like this and activates this pathway. So the more it activates this pathway, the more prostaglandins are released. The more prostaglandin is released, the more vasodilation and hypotension as well as fever. Clear? Now you understood what is the function of interleukin 1 and interleukin 6. Now instead of telling all this story, if I ask you a question, what is the function of interleukin 1 and interleukin 6, you have to tell they are responsible for hypotension and fever. Point number 2. Then what is what is TNF alpha? Tumor necrosis factor alpha. Let me tell you one thing, even tumor necrosis alpha also takes part in the same function. Right? It takes part in the same function. Right. Second important thing is, what is nitric oxide? Nitric oxide is responsible for hypotension. Okay, right. So these endotoxins perform the first thing successfully, that is activation of macrophage and causing all these things. The second important thing what these endotoxins do is that endotoxins will activate your complement system. Endotoxins, they will first activate an organ in your body, that is your liver. So when liver gets activated, your liver releases complement proteins. Your liver releases complement proteins. And what are these complement proteins? Complement proteins are C3A and C5A. These two are complement proteins. Fine. If these two are complement proteins, what is going to happen? What is going to happen is that these complement proteins, they act on another cell in my body, and this cell is called as mast cell. This cell is called as mast cell. What does mast cell consist of? All of you know, mast cell is consisting of what? Histamine. Right? Listen very carefully now. The more the endotoxin, the more activation of liver. The more the activation of liver, the more complement proteins are released. And these complement proteins, they make holes in the mast cell. They make holes in the mast cell. And what will happen when they make holes in the mast cell? Obviously, the histamine will be released out. So, there will be histamine release. There will be release of this histamine. So, all of you very well know histamine is responsible for causing two important things. What are those two important things, guys? One is called vasodilation. Another one is called as edema. Vasodilation and edema. Now you might get confused if the vessel, if this is the vessel previously, this is my blood vessel previously, and now my blood vessel has been dilated like this, then why there will be edema? Why there will be edema is that, see, this second picture is called vasodilation. Now the fluid within this will escape out. When the fluid within this will escape out, the fluid will stay outside the vessel, and that is what is called as edema, right? Full. Fine. Third important thing, what these endotoxins do is that, let me teach you one small concept. Then I'll tell you what is going to happen. We have something called a factor that is called as tissue factor. Right? So what this tissue factor basically does is that this tissue factor will convert factor 7 to 7A. 7A means activated factor 7. 7 is unactivated. 7A means activated. Now this 7A will in turn convert factor 10 to activated factor 10. Now this activated factor 10 will in turn convert PT. PT means prothrombin. It will in turn convert prothrombin to thrombin. And what does thrombin do? Yes, guys, tell me, what does thrombin do? All of you know, thrombin will convert fibrinogen. Yes, it will convert fibrinogen to fibrin. And what does fibrin do? Fibrin will form clots in your body whenever there is injury. Right? So this is basically called as blood coagulation cascade system. Right? So who is the starting factor here? That is tissue factor. If I produce some type of toxin and I start activating this tissue factor a lot, then what will happen? The more I activate the tissue factor, the more and more fibrin is released. The more I activate the tissue factor, the more and more concentrations of fibrin is released. So what will happen when there is increased concentration of fibrin? There will be clots everywhere in my body. And that is what is called as disseminated intravascular coagulation. So now I hope you understood that this endotoxin will activate your tissue factor. The more it activates your tissue factor, the more amount of fibrin is produced, the more clots in your body, and that is what is called as disseminated intravascular coagulation. So let us sum up all these things, guys. Let us sum up all these things. First thing, what does it do? It will activate your macrophage. Macrophage activation. So when your macrophage gets activated, what are the two important things which you can see? One is your fever. You see fever in the patient. Not only fever, you also see hypotension. Apart from that, second important thing is that it will even activate your complement system. It will even activate your complement system. So when it will activate your complement system, the second important thing it is going to cause is vasodilation. So obviously when there is vasodilation, you know there will be edema. Third important thing is that it will even activate your TF. TF means tissue factor. So when it will activate your tissue factor, what is going to happen? This will cause the release of what? Fibrin, and that will cause disseminated intravascular coagulation. So in a nutshell, if I wanted to discuss about all these three things, these three are the most important functions of what? Functions of endotoxin. So if you want to directly learn this box, which I'm drawing now, it is your wish. But if you want to learn with the concept, that would be better, because in your starting stages of your medicine, you always have to learn the concepts, right? Instead of knowing the important points, you have to learn the concepts and then know the important points, right? So these are the things which we have discussed today. So anyways, thank you so much guys. So this will be the end of the video.